The overall goal of this purification is to yield functional Helicobacter pylori neutrophil-activating protein, or H.pNAP, in its native form with high purity and high yield using negative mode batch chromatography with diethylaminoethyl ion exchange resin.
The main advantage of this technique is that H.pNAP can be purified in its native form in one step with high purity and high yield. Negative purification of H.pNAP starts with a batch binding procedure in which the soluble protein fraction from bacterial lysase is added to DEAE resin. The mixture is incubated to allow whole cell proteins to bind to the resin. H.pNAP present in the unbound fraction is then obtained by centrifugation or gravity flow.
The implications of this technique extend toward prevention, treatment, and prognosis of Helicobacter pylori-associated diseases, as well as cancer therapy. Because of the immunogenic and immunomodulatory properties of H.pNAP. Generally, individuals new to this method will struggle because H.pNAP easily aggregates during the cell lysis.
Demonstrating the procedure will be Zhi-Wei, a graduate student from my lab.
Working at 4 degrees Celsius, resuspend 50 milliliters of the cell pellet prepared as described in the text protocol in 20 milliliters of low-salt tris buffer at pH 9.0.